So let's now look at the cardiac muscle physiology. So before we go into that, I'm going to quickly review the skeletal muscle physiology. If you remember from ANP1, skeletal muscles have resting membrane potential, which is like inside of the membrane as compared to outside of the membrane. Inside is negative, outside is positive. Inside you have potassium ions, and outside you have sodium ions. And then we spoke about the axon terminal, which brings in the message. And once the neurotransmitter acetylcholine is released, it comes and sits on the ligand gates and opens up the sodium channels. And once the sodium comes inside the skeletal muscle, it becomes more positive than the outside. That is called as depolarization. While sodium moves in, potassium slowly goes out, making inside negative again. That is called repolarization. And here we see something that we did not see in skeletal muscle, but we saw in the neurons, that the potassium gates don't close in time and make the inside more negative than at rest. That is called as hyperpolarization. Hyperpolarization is a very important event that takes place in cardiac muscle cells. So this is important to understand. In skeletal muscle, the two major ions were sodium and potassium. And here we're going to talk about calcium as well. All right. In skeletal muscle, we spoke about calcium channels in the terminal cisternae and how they are released. They attach to the troponin, right, thereby releasing the tropomyosin to the myosin head to attach to the actin filament, right, and then the shortening takes place. All those events are the same here as well because we have the striations that we see in skeletal muscle. So we are not going to go into that those details because you already know it. So here we are going to talk about something a little different than what happens in skeletal muscle physiology. Do remember the skeletal muscles are voluntary and cardiac muscles are involuntary. So that's a big difference right there. Anyway, so and we also saw in skeletal muscle that the action potentials kind of ride one on top of the other, and they're stronger than the initial ones, so that they cause a wave summation and a tetanus, and that's when the shortening of the skeletal muscle takes place. So that is a big difference between skeletal muscle and cardiac muscle, that we do not have any tetanus in cardiac muscle. There's only one contraction, and that is a long, sustained contraction. While that contraction is taking place, you cannot have another action potential overriding it. Okay, so that is one big difference. So now let's go and look at some of the differences between cardiac muscle and skeletal muscle. Cardiac muscles are fairly short. Skeletal muscles are very long. They are semi-spindle shaped. They are cylindrical. These are branched, interconnected. And here we don't have branching in skeletal muscle. The cardiac muscles have intercalated discs with gap junctions. There is no tight binding, or there is no gap junctions in skeletal muscles. All the cells in the cardiac muscle, especially the chambers like the atria and the ventricles, they contract together. Okay, so it's almost like a domino effect where ions are moving from cell to cell to cell to cell, causing the contraction to take place together. That is called as a syncytium. Okay, whereas here in skeletal muscles, each muscle cell contracts on its own, though we have a concerted contraction of the muscle as a whole, and that is because of the individual muscle cells contracting to the neuron that is giving the message to them. Whereas here it is one single contraction, so they all kind of sync together and contract. They have 1 to 2 central nuclei. This is multinucleated. This cardiac muscle are highly vascular, meaning that every cell has to have oxygen supply at all times. Whereas here we don't need that vasculature because skeletal muscles can contract anaerobically as well. So they can produce lactic acid, and then that is taken back to the liver and converted back. reused. Whereas here we cannot have lactic acid production. Okay, so that is going to stop the heart. There are many mitochondria, 25% of the space, whereas in comparison, skeletal muscles have only 2%. They're all aerobic, whereas here you have aerobic and anaerobic respiration. And then T-tubules, myofibrils, that's fine. T-tubules are wider, fewer gap junctions. Here you have T-tubules at every sarcomere, right? So the ending of the Z disc, you have a T-tubule. Absolute refractory period is seen, meaning that once the action potential starts, another one cannot override it, right? So you do not have action potentials causing a wave summation. You have depolarization followed by repolarization, and then you can have the next action potential. So absolute refractory period is essential in cardiac muscle, whereas here you don't have absolute refractory period. You need tetanus in order for the skeletal muscles to contract. And here, if tetanus does occur, that is going to lead to fibrillation, meaning you don't have a proper electrical stimulus. Okay, so that is the heart attack happening. All right, so those are some of the similarities and dissimilarities we see between cardiac muscle and skeletal muscle. Now let's talk about some of the major differences, and let's focus on cardiac muscle now. Cardiac muscle works as one single unit. Okay, so this is called as functional syncytium, and it's made possible because of the presence of gap junctions between the cells, and the ions can move from cell to cell to cell to cell, and we will talk about that. So cardiac muscles have intercalated discs, desmosomes, and gap junctions. Okay, so and all three of them are very, very important. Cardiac muscle is innervated by intrinsic conduction system as opposed to stimulation by nerve ending as seen in skeletal muscle. So we don't have the axon. We do have axon terminal, but the main rhythm is set by pacemaker cells, which are part of the cardiac muscles themselves. This is called as autorrhythmicity. So we don't see the axon terminal, okay, innervating each and every skeletal muscle cell, right? So we don't have the muscle fiber and the axon terminal connection. There is no neuromuscular junction as such. Here we are talking about a group of cells which are going to trip over and over and over again. Okay, and that is what causes the action potential to stop. So that is called as autorrhythmicity. The absolute refractory period in which the sodium gates are closed and inactive is much more than in skeletal muscles. It lasts for 250 milliseconds as compared to 1 or 2 milliseconds in skeletal muscle. In fact, in skeletal muscles, we do not have refractory period, absolute refractory period after once the wave summation starts. And this is important. Having the absolute refractory period is essential for cardiac muscle. This prevents the tetanus and complete summation. Okay, we cannot have complete summation. So I can actually talk a little bit about this. So you have 250 milliseconds of depolarization and repolarization in contractile cells. And so following the depolarization, we see contraction. Following the repolarization, we are going to see relaxation. So the whole contraction relaxation kind of takes about 200 milliseconds. So it follows the action potential. So we'll talk about that. Membrane potential is unstable. Okay, so the pacemaker cells have to keep tripping, right? So it has to keep starting the action potential. That is called as an unstable membrane potential. We don't want it to come back to rest, right? Otherwise, we will rest for life. So this is called an unstable membrane potential. Again, we are going to talk more about this. These potentials are initiated by pacemaker cells. Okay? Unlike skeletal muscles, calcium ions play a very important role along with sodium and potassium. So in skeletal muscles, we saw that the action potential, that is depolarization and repolarization, is caused due to sodium and potassium, whereas in cardiac muscle cells, calcium plays equally, or in fact, more important role in the actual action potential. Okay, so forget about the part where it has to go and help in contraction. Yes, we do have that. But apart from that, calcium plays a very important role in actual depolarization of these cells. Okay. So there are two kinds of innervation conduction system, intrinsic and extrinsic. Extrinsic is the control of the rhythm, right? So when we have heartbeat, we do have like a normal heartbeat, and then when we are exercising, the heartbeat increases. When we are sleeping, the heartbeat decreases. So that increase and decrease in the heart rate or heartbeat is caused by the autonomic nervous system. So when sympathetic is turned on, the heartbeat increases. When parasympathetic is turned on, we have slower heart rate. But the normal beat, right? So like a constant beat is controlled by intrinsic conduction system. Okay, so the extrinsic is autonomic nervous system, but the intrinsic are the pacemaker cells, are also called as autorrhythmic cells. Okay, so in the heart there are two kinds of cells. One are the autorrhythmic cells, which form about 1% of the cardiac muscle cells, and the rest, 99%, are the contractile cells. The autorrhythmic cells are also called as non-contractile cells or pacemaker cells. So the only function of pacemaker cells, which is just that 1%, and they do not have contractile elements, is to actually start the rhythm. Okay, so that's the only function they have, and they do not need nervous conduction, but just to fine tune it. The action potential starts in these non-contractile autorhythmic pacemaker cells, and it spreads to other contractile cells via gap junctions. Okay, so these are the pacemaker cells. Okay, the unstable membrane potential. Okay, so of these cells causes the whole beating of the heart. Okay, so let's start with this diagram. So this is just for pacemaker cells. Okay, so how many? Just let's pause here. What percent of cells make the pacemaker cells? You guessed it right, 1%. Do they have contractile elements? No. What is their function? To keep the unstable membrane potential, to keep it going, to have the action potential. Okay, so they take over what the axon terminal is doing in skeletal muscle. So let's look at this. So at minus 60, the potassium gates close. Okay, at minus 40, that is the threshold. Okay, so that is the actual resting membrane potential. But we are starting from the previous cycle. The potassium channel closes, and this opens up the sodium gates. Okay, so this is called as slow sodium channels, very slow sodium channels, which makes it positive inside. So that is kind of depolarization, but this is called as pacemaker potential. This is a slow depolarization. At threshold, the sodium gates close, and that opens up calcium gates. So you have an influx of calcium. That is the actual depolarization. And at about 0, the calcium gates close, potassium channels open up. So potassiums are going out, making inside negative. And so you have inside being negative, and it doesn't stop at resting membrane, but it keeps going because it leads to hyperpolarization. Okay, so the potassium gates close very slowly, and this hyperpolarization is what causes the sodium gates to open. So hyperpolarization in the non-contractile autorhythmic cell is extremely important because that leads to the pacemaker potential. So if this were to remain open for a longer time, like if potassium were to, instead of stopping at minus 60, if it stopped at minus 80, for example, if it stayed longer, then it is going to make it a little slower. Okay, and that happens. And that happens when you have vagal tone or parasympathetic tone. And if sympathetic takes over, then this is going to be higher. Okay, so more calcium comes in, and that makes it contract a little bit more, and that's much faster. So just giving you an FYI here. Okay. So again, minus 60 is the hyperpolarized state where potassium gates close. This leads to the opening of sodium gates. That is slow depolarization, very slow sodium gates. So now we have sodium inside, right? Potassium is also inside, and then calcium comes in, and it is filled with calcium ions as well. And then potassium starts to go out. So as you have the inside building up with so much of positive ions, they're going to start leaking to the cells which are next door. Okay, so those, this is another same thing. So they start to leak to the surrounding cells, which are contractile cells, right? We have 99% of cardiac muscle cells, which are contractile cells. So they start to go into the next door, and that is enough for that positive ion. Okay, so the positive ions start leaking through gap junctions into the contractile cells, and that is enough to open up fast sodium gates. So sodium rushes in, okay, and makes it really positive. From minus 90, it goes to plus 30 very quickly, right? So depolarization takes place. This is for contractile cells. Depolarization takes place, and then it closes as suddenly as it opens, and there is a little lull where the depolarization stops, and there is a little fall, and at that time you have calcium coming in. Okay, so you have calcium coming in and potassium going out at the same time. So that is called as a plateau. Okay, so you have it kind of stagnates there, right? So you have like a plateau stage, and then around here calcium gates close, potassium is still open. So calcium is coming in, potassium is going out, calcium stops, potassium is still going out, and that leads to repolarization. Okay, now look at this. So as the sodium comes in, inside is positive, right? So calcium is released from the sarcoplasmic reticulum, that goes and attaches to the troponin, pulls the tropomyosin, and you have the contraction that is taking place. So as I was mentioning earlier, that these are contractile cells. Please do remember that. So as the depolarization takes, after depolarization, you have the contraction that is taking place. And after repolarization, you have the relaxation taking place. Okay, so you have like a sustained contraction, okay, a long contraction. And we are not talking about summation. There is no wave summation, no tetanus. It is just one action potential. This is one action potential, and you have one big contraction. So again, in short, let's summarize this quickly. So we have autorrhythmic cells or pacemaker cells, which constitute 1% of the cell, and the remaining 99% are the contractile cells. The pacemaker is the one which sets the tone, right? And you have a bunch of pacemaker cells, and we'll talk about that where they are present. So pacemaker cells are the ones which have unstable membrane potential. So they have slow sodium deep intake, which causes slow depolarization. That's the pacemaker potential. And then calcium ions come in to form the depolarized wave. Then it closes at 0, and then you have the calcium leaving and potassium leaving, causing repolarization followed by hyperpolarization. So hyperpolarization is very important because that is what sets the pacemaker cells, pacemaker potential. And as the pacemaker cells are filled with positive ions, they leak to the neighboring cells, causing them to have depolarization due to fast influx of sodium, and then the sodium gates close. Then you have slow calcium gates opening up, calcium comes in, and potassium going out. So you have a plateau. The longer the plateau, the more the contraction. Okay, that is your absolute refractory period. And then you have calcium gates closed, and then you have the um, potassium going up. Okay, I hope this makes sense. So please do watch animations of this. There are many animations online. I think McGraw-Hill also should have something. If not, just look online to see if you can find any animation. So this picture I really do like because this actually shows this is for contractile cells. This shows this sodium, and this is the electrical activity, and then showing the mechanical activity, that is a contraction relaxation. And this is the individual ions, right? So sodium comes in, closes. Then you have calcium coming in and then closing. And then you have potassium leaving the cell. So that's where you have that inverted graph. Okay. So when we look at the pacemaker cells, we have the pacemaker of the heart is called a sinoatrial node, okay, which is in the right, just at the base of superior vena cava. That is what sets the pace for the heart. It's about 72 beats per minute. And then at the junction of the atria and ventricle, you have another node, which is called as AV node. The whole lining or the pathway between the atria and ventricles are completely insulated. They do not have any opening. I'm talking about electrical signals, except for AV node. Okay, so that for that reason, AV node is really important. And then it comes down into the septum as AV bundle. Then the bundles branch. They divide. They're called as bundle branches, and then they come all the way to the apex, and then they go up as Purkinje fibers. So before they go into Purkinje fibers, they also innervate the papillary muscles. That is really important. Okay. So here we have the SA node fires, sends all the message to both the atria. Once it sends the message, right, the depolarized, then you have um, um, both the atria contracting after both the atria are depolarized. By that time, the message comes to the AV node. Okay, so the atria contract, and at that time you have the message going down, message meaning depolarized wave, going down the AV bundle, and then AV bundle bifurcates and then goes to the apex all the way to the side, right, to the whole wall. And before that, it innervates the papillary muscles, which makes these muscles contract, closing the valve. Okay, so we'll talk about that in the next part when we are going to put all of them together. So for now, all you have to know is what are the different nodes. So you have the SA node, AV node, AV bundle, also called as bundle of His, and then right and left bundle branches, and then you have Purkinje fibers and papillary muscles, which are also innervated. So the sequence of the events, it starts from the SA node, beats about 72 beats per minute under the influence of parasympathetic nervous system. Then it goes through AV node. Okay, so there is a little delay. It's almost like having 5 lanes come into 1 lane, stop, and then boom, it goes out into the AV bundle. AV bundle, it goes very fast because these are pretty big. Okay, these are big cells. And as I said earlier, there are no gap junctions between atria and ventricle. So AV bundle is the only connection. AV node is the only connection between SA node and the AV bundle. Then you have Purkinje fibers, which go to the walls superiorly. So they're innervating the whole ventricular wall. And then it innervates the papillary muscles just before the whole wall is innervated. Okay, so we will talk about that. So that is your pacemaker cells and contractile cells. Again, please watch some animation on YouTube or somewhere. Okay.